All right, so I know it's been a week of forgetting what we were doing, right? So hopefully you vaguely remember that we had started talking about energy and catabolism last time. So we went through some basic definitions, right, about how energy and entropy and how the Gibbs free energy tells you whether a reaction is likely to happen or not. Then we also talked about the different energy carriers and then activation energy. And then we finished just the beginning part of catabolism, like generally how things start to break down. And today we're going to go further into specifically glucose catabolism. But just as a review from last time to hopefully get you to think about what we did last time, can anybody tell me the answer? Sorry, I heard muted things. Now I'm hearing all different answers. Okay. The term blank refers to the heat energy, and whereas blank refers to disorder or randomness. I'm hearing B, I'm hearing C. Okay, so the term that refers to heat energy is enthalpy, right? And then the disorder is entropy, so it's B. So the next question. Good. All right, so enzymes help speed up reactions by lowering the activation energy. Just remember that just because the delta G is negative doesn't mean that it's going to happen in any meaningful time frame. So the enzymes are what helps us, or helps the cells make those reactions actually go faster. All right, so today we're going to get into glucose catabolism, and so the three main pathways. And then we'll start talking about what happens after catabolism, and whether or not you have a terminal electron acceptor present determines whether the cells will do fermentation versus going towards the citric acid cycle. All right, so let's get started with glucose catabolism. So last time we had basically talked about how the cell can break down the different food sources into molecules that it can start using in different metabolic pathways, right? So there are three main pathways for glucose catabolism. In the end, the result is that they all get broken down into two 3-carbon products, namely 2 molecules of pyruvate. And then each of the different pathways can generate different amounts of energy or transfers different amounts of electrons to the different energy carriers. And so we'll go into each one of these. But as you can see, they're all kind of interrelated, and we'll get into that too as we go on. All right, so just as something to kind of preface what we're talking about and make sure that what you want to pay attention to is to basically follow the carbons. So what's happening to glucose as it's getting broken down? When is ATP consumed? When is it generated? And then also when is the reducing power generated? And then of the 3 pathways, which ones generate more energy or more reducing power? So those are kind of the main takeaways that you want to get out of the different pathways. All right, so the first one we're going to talk about is glycolysis, or also known as the Embden-Meyerhoff-Parnas pathway. So this exists in bacteria, archaea, and eukaryotes. There are 10 steps that are broken down into 2 separate stages. The first stage is the investment stage. So you have to actually invest 2 ATP in order to break down the glucose, and then you're going to end up generating more ATP after that. But the cells do have to invest some energy to be able to prepare it to get broken up. Then in the second stage is the energy yielding phase. So this is where it actually gets split into 2 pyruvate. So throughout these different processes, so this is the same for all of them, we will generate ATP out of this, but this is via substrate level phosphorylation. So it's the transfer of a phosphoryl group from an organic substrate to make ATP. The other thing that I want you to pay attention to is that whether or not the reactions are reversible. So that's what amphibolic basically refers to, is so certain reactions in this pathway can participate both in the breakdown and in the synthesis, so going the other way, which we will talk about maybe in a couple of lectures. So just you want to pay attention to which reactions are reversible. So I'll start talking about some of the different things here. So this is the overview of what glycolysis or the EMP pathway looks like. So again, it's split into two stages, right? So the first stage here is where you're putting into ATP to get it ready to be to split, and then here you're actually generating the 2-pyruvate, and you end up generating more ATP and then the reducing power. So some of the things that you want to take a look at, and just things to know in general, right? So again, a lot of these reactions, so you'll notice the ones that, so these arrows here means that they're reversible, the ones that go in both directions. So in that sense, those are the ones that can be used both in the synthesis pathway that we'll learn about later, and then in the breakdown. The ones, of course, with the single arrows like these are the irreversible ones. These are the ones that are key to whether it's breaking down or synthesizing. And so, as you can imagine, these are also the irreversible steps are the ones that are also key for the cell to regulate. So it's key for regulation. And one of this reaction here actually is one of the key steps for regulation, in the sense that the cell will basically want to balance when it needs energy or when it needs to be building biomass, right? And so when the cell needs energy, this phosphofructokinase, this is the one that is going to push the pathways down to breaking down instead of synthesizing. Okay, so this kind of looks like a mess. So what we're going to do is I'm just going to break it down for you, and we'll just go through it step by step. And I'll kind of just point out the major things and just kind of what I mean by following the carbons and how all of that will work out. Okay, so, all right, so we're gonna start, we'll just split this. So for stage 1, again, we start with glucose. And the first step is an irreversible step, so it goes, and so it requires energy. And you end up with this glucose 6-phosphate. So this is a good one to remember, because this one it'll show up in the other pathways too. So just things that you want to be aware of. And then this we'll go through after a couple of steps. So we'll again, this is your next irreversible step that goes to fructose 1,6-bisphosphate. Okay, so why don't I finish the first pathway first, and then we'll. So again, there's a few more steps, and you end up with, so at the end of stage 1, you end up with make sure I'm spelling these things. Glyceraldehyde 3-phosphate. So this part here, you end up actually, we'll often refer to this as G3P, but you end up with 2 of these. So this gets split here. So the basic thing that you want to do is, glucose starts off with 6 carbons, right? So after the first one, what's basically happening in the beginning is that it needs to get ready for you to split it. And so you're going to use up the first this first ATP, and you're going to end up attaching it to one end, to the 6 end of the glucose. And then at the other stage where you are going to use the ATP, you're going to add the phosphate to the 1 carbon. So again, you're going to use one more ATP here to add the phosphate groups here. And then at the end, you'll end up with essentially 2 3-carbon molecules that both have a phosphate on one end, right? So at the end of stage 1, you've used 2 ATP. Okay, so now for stage 2, so it's basically everything times 2. Because now you have 2 of these, so each one of the G3Ps are going to go through stage 2 separately. So you have G3P, and now, so basically now I'm just going to write out the steps where the energy and the reducing power come out. And so at this step here, you end up with 1 NADH here, and this is 1,3 bisphosphoglycerate. You can look up what my scribbles are or what the actual names are, but the key is to just know where the energy is coming out. And so in this step here, so this is the first place where we're actually getting out ATP. So in the beginning, so from G3P to this first one, you still end up with the 3 carbons with the phosphate, but now here, so here you see we're making 1 ATP, so one of the phosphates get used up. You donate one of those phosphates to ADP to get the actual ATP. And so on this end, you'll just have ones that have the phosphate on the 3 carbon. And then it goes through a couple more steps where you end up with phosphoenolpyruvate, so you end up at PEP. And in this case, what's happening here is you end up with a few rearrangements, and so the phosphate actually gets moved to the 2nd carbon. And then finally, you get to pyruvate. And your phosphates are gone, but now you have pyruvate. So here we've generated ATP here, ATP here. We've also generated NADH here. And so in stage 2, so stage 2 you get +2 NADH and +4 ATP, because remember this has to happen for each. So I've drawn out all the carbons on this side, but I haven't drawn out that this is ×2. So just remember that each one of these you get generated ×2. So on this end you get 2 NADH and then 4 ATP. So in the end you have a net. In the end you have a net of 2 ATP and 2 NADH, plus the 2 pyruvate. All right?
So 2 ATP, 2 NADH, and pyruvate?
Yep. That's the total, the net from the EMP pathway. Yep.
Do we have to memorize the signs associated with each step?
Nope. All right. All of this is, I've just rewritten exactly what was written on in the picture on the slide. I didn't write any new information on here. This is just as a review to show you. Yep. Sorry, what? Okay, so we were talking about glycolysis here, but there's another pathway. So the second pathway we're going to talk about is this Ender-Duderoff pathway. So in this pathway, again, this is just a different route to catabolizing sugars. It's, again, used by bacteria, archaea, and eukaryotes. So this one is actually key for the bacteria that are living in your gut. So this allows enteric bacteria to use intestinal mucus. So your intestines, the mucus from your intestines create what are called sugar acids. And so this pathway here is what allows those bacteria to actually, instead of just getting glucose, they will get them in form of these sugar acids, and this is how those bacteria can take advantage of that and generate energy and things like that way. This pathway is also thought that it had maybe evolved earlier because it has few steps, generates fewer ATP, but it's also found in a wider range of prokaryotes. So like I said, this shortcuts the EMP pathway, yielding only half the energy. So in the end, it generates 1 NADPH, that's different, right? And then 1 NADH. So we'll look, sorry. What, sorry? That pathway. So, yeah. Okay, so again, so in this pathway, again, like I said, if you notice, the first step is the same, right? So you end up with this glucose 6-phosphate. And then the key next step is that you generate this, the difference is that you're gonna get this NADPH versus the NADH. And then that turns this into this 6P, this gluconolactone. So these two here, the lactanates, those are the sugar acids. And so that's where actually you can actually just have for the enteric bacteria can actually just enter things at this step, and they don't have to enter them as glucose. But the pathway is written out as if you had an alternative way of just handling the glucose itself. And then what you'll see, and I'll write this one out again. So it goes through several different steps this time, and in the end, you do get 1 pyruvate, but then you also get 1 G3P. So this time the pathway, it has its own step to generate 1 pyruvate, but it generates 1 G3P, which you then run through stage 2 of the EMP pathway, and you get pyruvate at the end of that one, that way. All right, so I'll do the same thing. We'll go through just the summary of this. Oops, sorry. That's these guys here. So this is the sugar acid, so that's different. The ED pathway. So again, we're starting with we're starting with glucose. And this is the same step, right? So you get the glucose 6-phosphate. So here you're using this ATP, but this is the exact same first step. So now this is where the ED pathway is different. So this step here where you go and you put in the NADP, and you end up with NADPH. And then there's another step and you end up with the this 2-oxo-3-deoxy-6-phosphogluconate. So this one, I'm pointing this step out because this is where you end up splitting the molecule here. So at this point, you end up splitting into pyruvate and G3P. Right, so pyruvate is kind of always what the end goal of these pathways is what we're looking for, right? And then this can go through stage 2 of glycolysis. And so in the end, of course, this you get the 1 pyruvate here. So here you're generating this energy here, right? This reducing power here, but you end up with the 1 pyruvate coming out of here. And then now if we were to, so what happened? So coming out of here, essentially is just 1 arm of the stage 2, and so you end up with 1 NADH and 2 ATP. Right? So if you imagine only going through 1 time, because there's only 1 G3P molecule to go through stage 2, so you only get the 1 NADH and the 2 ATP. So now if you balance this out with what we used here and here, so the total net for this pathway is just 1 ATP, but you end up with 1 NADH and 1 NADPH. Right, because these 2 here are positive, and then plus the 1 ATP that we have to take in. So you do, again, so you end up not generating as much ATP, right? Because you generate more ATP going through glycolysis stage 2 than you do, because this one just, all we generated here was this NADPH and it went straight to pyruvate already, right? So we lost all this energy created here. Is the one from the ED pathways. All right, so the last pathway is the pentose phosphate pathway. So again, this one, so the main thing about this one is it generates the possibility for all these different intermediates. So the key intermediate here is this ribulose 5-phosphate. So that one, so this is a key intermediate that will allow you to make different amino acids, vitamins, because all of this can be redirected for biosynthesis of different sized sugars. So this one, again, so it's very similar, right? So it starts off with the same way that the ED pathway does. So you get to that 6-phosphogluconate. So you get here the same way. But then there's a second oxidation of NADP plus, and then so that is how when you lose that carbon dioxide, so that's how you get from a 6 to 5 carbon sugar. And then from there, there's all these possibilities of what kinds of different sugar molecules you can get. But basically, if the sugars aren't needed at that time in the cell, then you can regenerate fructose 6-phosphate and then pass that back into glycolysis.
Is 6-phosphogluconate the same as glucose 6-phosphate?
No, the 6-phosphogluconate is the, so this is what the pathway looks like. So here, again, this is, again, remember, because it's the same as the ED pathway, everything goes through glucose 6-phosphate, right? But then it goes to, in the ED pathway, you get to this, the lactone and then the 6-phosphogluconate. But now here, so again, what we need to do is lose one carbon from here, and so this is the step where you get one more NADPH in this pathway, and then you end up with this ribulose 5-phosphate. And so from here, like I said, it can do many different reactions and end up with all these different sugars depending on what the cell needs at that point and if it needs to be doing biosynthesis of something. So this pathway is kind of more key in the sense that if it needs to do synthesis.
Where in the mitochondria is this all occurring?
We are not talking about mitochondria, so we're talking about bacteria. So this is all in the cytoplasm. So there's no compartments, right, in the bacteria. So all of these are just for E. coli. So we're not talking about. Later on, we'll talk about more things when we talk about photosynthesis and things like that. But right now, we're just talking about E. coli as an example. So all this is in the cytoplasm. Okay, so for this one, like I said, we're not going to go through the tracking, but just to know that the key is that you lose this one carbon to get to this the ribose 5-phosphate. Okay, so again, so these are 3 different possible pathways for catabolism of glucose. And remember that they're all interconnected, and you end up with different amounts of ATP and different reducing power at the end of these. So these are the key things that you want to be able to know about the different pathways. But now, just because we've gotten to pyruvate, that doesn't mean that's the end, right? So we've essentially generated all of this reducing power. But as we talked about with the different energy carriers, they're just supposed to be reused and just kind of transporting electrons. We need some way to regenerate the NAD+ for the cells to be able to use that up again. So the first thing we'll mention is fermentation. So this is in the absence of oxygen or an alternative terminal electron acceptor. So we're going to talk about in a second, and next time we're going to talk about the citric acid cycle and the electron transport chain. But in the case where you don't actually have a terminal electron acceptor, cells can do fermentation. And so the electrons are transferred to products of pyruvate. So again, you have pyruvate, and so what's going to happen is it's going to use the pyruvate, and then it's going to use the NADH and transfer the electrons in some form back to the pyruvate. And in the end, you end up with a net balance of oxygen and hydrogen in reference to glucose. So there are different types of fermentation that you can have. So in essence, it goes from 2-pyruvate to one of these options. So in lactate fermentation, you get 2-lactate. In ethanolic fermentation, you get ethanol and carbon dioxide. In heterolactic fermentation, you can get 1 lactate and 1 ethanol. And then you can have also mixed acid fermentation. So you get the acetate, formate, lactate, succinate, ethanol, hydrogen, and carbon dioxide gas. So there are many different pathways. It kind of just depends on the bacteria that you're looking at. And this is just kind of a summary of what those different pathways are. In the end, you're not generating as much energy. So the energy that you're getting out of it is the energy that we got from, let's say it went through glycolysis. So that's the energy that we're getting out of it. And so it's not going to generate as much energy as if it has a terminal electron acceptor, which we'll see soon. But so in this case, if it needs more energy, it's just going to eat up a lot more of the food source that it has. And in the end, it doesn't really do anything with these, right? So these end products are kind of just waste to the cell. It doesn't necessarily do something with them. If it's living in a community, maybe some other bacteria might use it, but usually it doesn't really do anything with it. But we have learned to do things with these end products, right? So we can make alcoholic beverages from them, do yogurt, cheese, and butter from some of these, and then also just kind of industrial chemicals that we can use, that you can, instead of using the petroleum-based processes, people have shifted to bacterial fermentation processes to produce some of these chemicals. All right, so the other option is if there's an electron acceptor available, right? So, of course, if there's an electron acceptor available, it's going to go through the tricarboxylic acid cycle, so the TCA cycle, because it's going to generate more energy that way. And so we'll talk about the TCA cycle next. So there are 10 known variations of the TCA cycle, all happening in diverse species of bacteria. So you might also heard this as the Krebs cycle. So that's the most common known one. It's found in most bacteria and eukaryotes. So if you remember now and I'll show up a picture in the cycle and it'll be a little bit more clear. So we ended it at pyruvate, right? But what enters into the TCA cycle is actually acetyl-CoA. And so what needs to happen is an oxidative decarboxylation step, which removes one carbon. So it goes from the 3-carbon molecule to a 2-carbon molecule. And so this removal of the carbon, and then it transfers 2 electrons to NAD+. So this is done by, so this is a key enzyme in this process called the pyruvate dehydrogenase complex. And so this is what does this process and enables things to enter the TCA cycle. But remember, we talked about last time, we were just talking about the general catabolism of other molecules. So glucose is not the only thing, right? We had talked about lipids, peptides, and all of that. And all of those in the end ended up looking like something that could be converted to acetyl-CoA. And so all of those things can now enter the TCA cycle here. So this is what the whole thing looks like. And again, so here's the key. So again, remembering that all the different things, the food sources that can be broken down, right, can enter. So we had just talked about the glucose version, and so how pyruvate, what that journey looked like. But remember, we had talked about different other different molecules that get broken down and eventually look like acetyl-CoA. So again, it's a cycle, right? So that means it's circular. And so it's going to go in through here. It's going to combine with this oxaloacetate, and it's going to go through a bunch of steps. Some steps in which you're going to lose carbons, and some steps in which you're going to gain energy, gain reducing power. And so those are the kind of the things that we want to pay attention to. But some of these, just, you know, things that you might have seen before, just some of the different sugar names. Again, in other situations, when the cell needs to go through synthesis, it might need to take some of these intermediates away, and just kind of remember how that might affect whether one reaction is happening or not when you change the concentrations of reactants and products. Okay, so this is the part that I want you to care about. So here, what we'll do is we'll start tracking the carbons here. So so this here, is, remember, we're starting with pyruvate, right? This has the 3 carbons. And so this is the key, the first step that will take it to acetyl-CoA, right? This is that first one, but it already loses 1 carbon here. So now we're at 2 carbons. Okay, so this is going to actually combine with this oxaloacetate. So, but in reality, let's make this a different color just so we can track this. So here we're going to have the acetyl-CoA and the oxaloacetate combining, and so you're going to get this 6-carbon molecule from which is the citrate, which is the 6-carbon molecule from these two coming together, from the oxaloacetate and the acetyl-CoA coming together. So then what happens is you lose a carbon, but which carbon is it going to lose? Which one is it going to come from? So scientists have actually done experiments to figure out which carbon is getting lost, and so they've done these with essentially carbon-14 molecules, and so you do put in these radio-labeled molecules, and you can figure out which one is which just by tracing the radioactivity. And so what they figured out is that this first CO2 that's lost here is one of the ones that, oops, sorry, the wrong color. Sorry about that. It's actually one of the ones that are coming from the oxaloacetate. And so what this one looks like, the 5 carbons from here, looks like this. And then again, this one, the next carbon dioxide, they did again with the tracer experiments, showed that again, it's a carbon coming from the original oxaloacetate. And so you end up, in succinyl-CoA, you end up with 2 carbons from each of these. When you, at this step here, right here, you end up with succinate. But at this point, the carbons, it's a symmetrical compound, and you can't really tell, and so the carbons are rearranged, and now it's kind of just, you reassociate those carbons as it doesn't, you can't really tell which one it's coming from at that point. And so now all we really cared about is that which ones had gotten lost here and here. And so eventually, right, it's a cycle, so it's going to go back in, and you won't be able to tell at this point. And so again, they all become oxaloacetate carbons. But again, just note that you are getting NADH, right, out of this. You're getting ATP. So some of you might have learned this before as GTP. The book focuses on the E. coli version, and so depending on what organism you're studying, you might have learned this as GTP, and that's also correct. It's just the book is going specifically over the E. coli pathway. And then you get FADH2 out of here and NADH out of here, right? So general accounting, so what you end up with in general, so per turn, so each time around, you're going to end up with 3 CO2, 1 ATP, 4 NADH, and 1 FADH2. But per glucose, just because we like to account per glucose starting from glycolysis, so remember you're going to end up with 2 pyruvates going through each time. And so it's just double. So you'll get 6, 2, 8, and 2 of these. All right, so just in general, the cell, and we'll learn about this next time, but what happens is all of this reducing power has a way of getting turned back into ATP, right? And so the theoretical, in general, again, so per glucose, you end up with 6 carbon dioxide, 2 ATP, 8 NADH, and 2 FADH2. So now there are numbers, and oh, sorry, go ahead. Yep. Theoretical yields of how this happens, and so for each, you get theoretically 3 ATP per NADH and 2 ATP per FADH2. And so from one glucose, so just for the TCA cycle itself, you get 2 ATP out of the cycle itself, right? You get 3 ATP per 8 NADH, and then 2 ATP per 2 FADH2. So that's 30 ATP just from the TCA cycle. And then you also got this, we're trying to count per glucose, right? So you had already gotten 2 ATP from glycolysis and 2 NADH from my glycolysis, and so that's another 8 ATP. So a total theoretical maximum ATP per glucose, you can get 38. But in reality, the cell yields about 20, right? The cell's doing so many other things that it's not going to just be focusing only on every single glucose's like maximum yield of ATP. And so it has to use some of it here, some of it to do something else. And so in the end, in reality, it gets about 20 ATP for this process. Okay, so the last thing we'll go through today is this glyoxylate bypass. And so some organisms, they can use alternate pathways. So this is in the case if the cell is in a situation where it doesn't want to prevent any loss of carbon. So the carbon dioxide that gets thrown out at the end of these, that's just a loss of carbon for the cell. And so if it's in a situation where it needs a little bit of energy, but then it can't waste the carbons, it actually needs to start synthesizing things. Some of these cells can go and divert isocitrate into succinate and glyoxylate. So this glyoxylate here, succinate is already part of the cycle. So in the end, so the glyoxylate also then goes straight into malate here, and so, but you don't lose the different the carbons here, and so it can, if it needs to, it can start synthesizing from those carbons there. So the net reaction is just written here. So you get 2-acetyl-CoA plus oxaloacetate, plus 1 NAD plus, you'll get succinate, malate, 2 coenzyme A's, and then the NADH. So again, you're not generating as much energy out of this because you're skipping the steps around here, but this is a way for the cell to save carbons if it needs to. All right, that's it. So next time we'll figure out what happens with the NADH and we'll go over the electron transfer chain and proton motive force.